In this video, we are going to explore in some depth the diverse and interesting biology behind retinitis pigmentosa. In the companion video, we cover the clinical aspects of RP, age of onset, the effects on vision, how it progresses, and how to test for it. Retinitis pigmentosa is a disease of the retina that affects about 1 in 4,000 people. More specifically, RP is one of several kinds of retinal dystrophy, meaning it is a degeneration of the cells from internal genetic/functional defects. They self-destruct. These degenerations can affect either rod or cone cells. RP is the best known example of rod cell degeneration. In recent videos, we have explored the complicated structure and internal workings of the photoreceptor. For example, phototransduction, the visual cycle, and protein trafficking. The list of causes of RP is as long as the list of processes needed to keep the rod cell functioning. Why do cells die? Indeed, there is a lot in that answer. Photoreceptor cells are very complicated machines. Defects can occur in any one of many working parts. And I would add, the answer is far from being completely known. We will start with a brief review of the clinical effects, so you can see how they result from the biology/pathology. Picking an average case, someone who notices visual defects beginning in the late teens or early 20s. A colorful daytime scene on the upper left, and your perception on the lower right. Since this is in bright light, the colorful image is formed by the cone cells. At nighttime, in low light conditions, the image is formed by rod cells. They don't perceive color, so you get a grayscale image. The fovea is the center of the retina, and it is where the cones are most densely packed. Everything peripheral to that quickly becomes mostly rods. In RP, rod cells are the first to die, so the first vision effect is loss of peripheral vision at night, which increases gradually but progressively over time. Cone cells do not go unaffected. Rod cells, it turns out, have an important function in supporting cone cell function. As a consequence, as the rod cell population declines, cone cells are also lost just later. So, at some medium time point, it becomes difficult to function in low light situations, and at a later time point, it also becomes difficult to function even in daytime. On office testing, visual acuity is reduced, and a visual field shows reduced peripheral vision. A special electrical test called an ERG is a key tool used to diagnose which retinal problem and to follow its progress. Eventually, retinal appearance typically evolves from normal to this. The most notable feature is the pigment clumps. In addition, there is pigment loss, the arterioles are narrowed, and the disc is somewhat pale. This is advanced RP with full effects on vision. Diagnosis based on the symptoms and physical appearance goes back to the 1800s. It was not until the age of molecular biology that anyone began to figure out the causes. This 1990 report in Nature represents the beginning of a new era in RP research. It is the first report of a specific genetic defect in rhodopsin, the pigment that senses light in the rod cells, that is mapped to a specific chromosomal location, and the specific physiologic defect identified. In this report, they identified a mutation in the long arm of chromosome 3. In codon 23, instead of the normal C, cytosine, there is a substitution of an A, adenine. That change in the genetic code causes an amino acid substitution of histidine for the normal proline. And so the normal structure and function of this rhodopsin is changed. The rod cells suffer, and RP results. And here we have the first entry into the modern biology of RP. Here is what that mutation means. Among the 23 pairs of chromosomes we have, the rhodopsin gene is located on the long arm of chromosome 3. This shows the banding pattern on the long arm of that chromosome and the address of the Rho gene. Expanding that location shows the 5 exons, intervening introns, and untranslated regions that altogether make up the Rho gene. Going in one more step shows the base pairs that make up the gene. We are just showing the base pairs from exon 1. Within the cell nucleus, the DNA base pair sequence is transcribed onto pre-mRNA. The introns are excised by the spliceosome. The remaining base pair sequence is the mature mRNA, which is then translated into the amino acid sequence that makes up the protein. Other forces then take over, causing the protein to fold into its final functional form. Looking closer at the Rho gene, the blue print designates the translated portion. The other sequences may have other functions like controller regions, but they are not translated. Almost all genes begin translation with ATG, the methionine codon. Here are the first 6 codons and their corresponding amino acids. Here you are looking at the heart of the system, the translation of genetic code from nucleic acid codons to amino acid sequence. Here I have circled codon number 23, which reads CCC, coding for the amino acid proline. In the specific mutation we are talking about, the middle base is changed from C to A, changing CCC to CAC, thereby changing the amino acid from proline to histidine. This change has a significant effect on the chemical properties, ergo its function. And so we see the first defined defect. 5 years later, in 1995, 15 genes had been potentially identified, 5 of which had been specifically identified. But for the most part, their functions were still being worked out. From there on, a lot has happened in the last 20 years. There are several ways of looking at RP. Mode of inheritance is one. In about half of cases, a specific mode of inheritance can be identified: dominant, recessive, or X-linked. There are additional rare modes, mitochondrial and digenic, we will not cover here. Notice that in about half of cases the mode of inheritance cannot be identified. This is a list of all the RP versions that are inherited as autosomal dominant, here listed by version number. There are too many to discuss all of them, so we will pick the 5 most common. Number 1 on our list is rhodopsin. The most common cause of autosomal dominant RP is a defect in the Rho gene, responsible for 20 to 30% of autosomal dominant cases. Any and all mutations in the Rho gene are categorized as RP number 4. Since rhodopsin is the molecule responsible for sensing light, this does seem like a key place. Within a rod cell, the molecule rhodopsin with attached retinal is the G protein coupled receptor that senses the arrival of a photon of light and then triggers the phototransduction cascade in rod cells. There are more than 100 documented Rho mutations, meaning there are a variety of different effects. Here is a good place to ask the next logical question. When there is a mutation, what harm does it do? How does it affect cell function? Why do cells die? This is a deep question that to date has limited understanding. Regarding the current example, rhodopsin, since the shape of a protein is key to its function, even small changes in shape could cause misfolding or change in chemical properties causing loss of function. True as far as it goes, but there's more. For rhodopsin, Mendez et al. Groups the possible causes of dysfunction and death into 7 categories. Let's look at some of his examples. In Mendez class 1, the protein is not properly transported within the cell. it does not get from the inner segment to the outer segment. Note that only class 2 is misfolding, here causing the protein to become stuck in the endoplasmic reticulum after translation. Cells have a mechanism for identifying and disposing of misfolded proteins, the UPR, or unfolded protein response. Accumulation of extra proteins stresses the endoplasmic reticulum, potentially causing cell death. Interestingly, there are mutations that cause rhodopsin to be continuously activated. It seems the brain disregards a continuous on signal, and the effect is the same as if there were no rod signals. This is called congenital stationary night blindness, which results in night blindness from youth, but it does not progress. This is the OMIM list of diseases caused by rhodopsin mutations, showing they can result in more than one disease and show different modes of inheritance. Nothing in RP is simple. The second most common cause of autosomal dominant RP is a defect in the spliceosome, specifically in PRPF, pre-mRNA processing factor 31. This takes us into a relatively new area of biology, the spliceosome. Briefly stated, when DNA is transcribed into pre-mRNA in the nucleus, the pre-mRNA contains extra regions which are not functional in the final mRNA product. Exons are the useful parts which are retained, introns on the parts that are removed, that are spliced out before the mRNA leaves the nucleus. It also serves the function of allowing different exons to be combined in different ways. Several proteins can be put together from a single set of instructions. Multiple defects have been associated with RP. The most common one is PRPF 31. Located on the long arm of chromosome 19, it is in an important part of the spliceosome complex, required for the assembly of the U456 component. Within the spliceosome, mutations can also occur in several other PRPFs listed here. The third most common defect involves a membrane protein, peripherin, abbreviated PRPH. Similar initials to PRPF, but quite different in function. Historically, this gene was originally called RDS, retinal degeneration slow. Eventually, it was found to be peripherin, which forms a four-part complex with ROM, retinal outer segment membrane protein 1. This wider view shows the peripherin-ROM complex is important for creating and stabilizing the end of the disc. It also connects to a GARP protein, which in turn connects to the CNG channel in the plasma membrane, thus establishing disc spacing. This is the OMIM list of the multiple retinal problems that result from PRPH defects. If you want to look this up, the gene name is PRPH2, not to be confused with PRPH. RP1 was the fourth gene to be identified with autosomal dominant RP. When this gene was discovered as associated with RP, the first thing that was known was its activity was controlled by oxygen level in the retina. So it was named ORP1 for oxygen-regulated photoreceptor protein 1. More recently, it was found that this gene codes for a member of the double cortin family. This is a two-part protein that regulates microtubule growth and assembly. It is very important in growing neurons in addition to photoreceptor cells. Number 5 on our list is IMPDH1. It is an enzyme important for the synthesis of the purine nucleotides adenine and guanine. It is the rate-limiting step in their synthesis, so defects here affect many cellular functions which depend on these nucleotides. This is one view of purine metabolism, selectively excerpted from the full KEGG diagram. The core of protein metabolism that we're interested in is easier to understand in this further simplified diagram. Synthesis of the two purine nucleotides starts with PRPP, which through 10 steps becomes inosine monophosphate, the common origin from which both adenosine and guanosine are synthesized. IMPDH1 controls the synthesis of GMP. Note the last reaction pathway requires ATP, while the adenosine pathway requires GTP. In normal cells, this neat little cross supply regulates the synthesis of each so that they are produced in nearly equal quantities. However, if IMPDH is lacking or defective, you cannot produce guanosine. Then you cannot produce adenosine. Given how important these are in cellular functions, you can see how much problem it is if you cannot produce them. One additional comment. The IMPDH1 gene is complicated. It has 18 exons and 8 different splice variants. Relatively recent work suggests one of the isoforms is particular to the retina. That is significant because one of the research questions of our time is why you can have a defect in such a significant gene and not have the entire organism fail. Now we move to the list of autosomal recessive RP varieties. These tend to be the more severe form of the disease. Again, there are too many to cover them all, so we will discuss the top 6. Number 1 is USH2A, the Usher gene. Usher syndrome was covered in the clinical video. Basically, it is a syndrome in which the RPI changes occur associated with hearing and balance defects. Multiple genes are known to cause various forms of Usher syndrome. The question is, how can genes that affect vision also affect hearing? What do they have in common? The answer is a bit complicated, so we will move the discussion of this and Bardet-Biedl syndrome to later in the video. ABCA4 is a transport protein located in the outer segment disc. In this more magnified view of the disc, we see the arrival of light causing 11-cis retinal to change into all-trans retinal, most of which goes into the cytoplasm of the cell, where the waiting retinal dehydrogenase turns it into all-trans retinol, and it enters the visual cycle. But some of the all-trans retinal goes inside the disc, technically the intradiscal space. There it forms a complex with phosphatidylethynolamine, or PE. ABCA4 is part of a large family of transport proteins. In this case, its role is the transport of all-trans retinal from the intradiscal space into the cytoplasm. Here it gets the mildly colorful description of flip-ace. Defective ABCA4 means all-trans retinal can't escape and is complexed with A2PE. Both of these compounds are known to be toxic to RPE cells. How does that happen? Because eventually the outer segments/discs of the photoreceptors are digested by RPE cells, and thus they ingest those substances. A2PE becomes A2E, the specific offender. To be complete, there is additional recent research suggesting accumulation of 11-cis retinal is also a problem. ABCA4 has been found to show additional activity in changing the 11-cis isomer to the all-trans isomer. How significant is that? This is a list of the multiple diseases caused by ABCA4 transporter defect. Now, with phosphodiesterase, we have come to the familiar territory of the phototransduction cascade. Phosphodiesterase is the enzyme that breaks down cyclic GMP. It has 4 subunits. Alpha and beta are the active enzymes with the gamma subunit attached to each, which regulates their activity. Activated transducin binds the gamma subunits and allows the alpha and beta to become active. If PDE is not working, cyclic GMP builds up, causing the sensitive systems in the cell to go haywire and die. These are the diseases caused by phosphodiesterase mutations. Genetic defects have been mapped to each PDE subunit. PDE6A mutation causes RP43, PDE6B mutation causes RP40 and congenital stationary night blindness. RPE65 brings us to the visual cycle, the pathway that is responsible for regenerating active 11-cis retinal after exposure to light. The enzymes highlighted in pink have been found to be defective in different versions of RP. There are multiple potential defects in this pathway. We will not cover them all here. The severity of the effects is based on the redundancy of the system. For example, there are multiple versions of the retinal dehydrogenase, but only one enzyme that converts all trans-retinal esters to an 11-cis retinol, that is RPE65. Therefore, defects in this enzyme have more severe consequences. Remember, the CNG channel is located in the plasma membrane of the photoreceptor. It is a gate that controls how much calcium flows into the photoreceptor outer segment based on the concentration of cyclic GMP. It is key to the cell's function. This concept view shows it has 4 subunits, and a more detailed view of the parts. If the CNG channel is not functional, then calcium level is uncontrolled and the cell doesn't function. Just for fun, I have shown both rod and cone versions here. When the cones are defective, then color vision is lost, thus achromatopsia. X-linked versions come in 2 main types, RP2 and RPGR. RPGR, or Retinitis Pigmentosa GTPase regulator. Mutations in this gene are by far the most common cause of X-linked RP, accounting for about 3/4 of the cases. While it is the case that the full role of RPGR is still being investigated, there has been a lot of work put into this, and it deserves a bit of extra attention. The RPGR gene is expressed in multiple tissues. With 19 exons in the human, there are 10 splice variants, 2 of which are particularly important to us. This is an expanded view of the gene map, showing significant differences. Let's look closer. Here is the gene map, showing the location of all 19 RPGR exons. This is to remind you that the spliceosome takes the pre-mRNA exons and can alternatively splice them into multiple combinations. If that sounded surprising when we mentioned it before, we are now at a real-life example. The RPGR gene can be spliced into these 2 main isoforms. The first one, EX1 to 19, is called the constitutive one and is expressed in cells throughout the body. The second is noted as ORF15, present only in mature photoreceptor cells. The protein domain maps show both have segments from exons 1 through 13. In the first isoform, the 14/15 segment is skipped, ending with 16 to 19. In the second isoform, exons 1 to 14 are included, followed by a uniquely spliced exon 15. The ending 16 through 19 segments are not included. As we mentioned a moment ago, the isoform X1 through 19 is located throughout the body. It is also present in the photoreceptor cell during development. However, as photoreceptor cells mature, they stop producing the first isoform and switch to production of the ORF15. It ends up the ORF15 form is exclusive to photoreceptor cells. Mutations are spread out in the exons 1 through 13. The ORF15 region is the hotspot. There are no pathological mutations in exons 16 through 19. RPGR is located mostly in the connecting cilium. What does it do there? It appears to be carrying on relationships with multiple other proteins, its interactome. First off, it appears to be held in place by RP-GRIP1, RPGR interacting protein. RP-GRIP serves as a scaffold attaching the RPGR protein to the plasma membrane, holding it in the cilia region. That puts it in a position to affect protein trafficking from the inner segment to the outer segment. It also interacts with multiple other important proteins listed here. Its role in these interactions is still being worked out. As I said a minute ago, in addition to being a major cause of RP, this protein is situated in an interesting functional location and generates a significant amount of research interest. RP2, the next entry in our RP series, is no less interesting. This connects to the world of G protein signaling, of which we have seen one example in phototransduction, and we are about to see a large and important addition to this world. We digress a moment for the big picture. The systems that use adenosine are mostly organized for energy use and storage. Systems that use guanosine are mostly controllers or switches. For one example, from phototransduction, transducin was a G protein in 3 parts. The alpha unit was activated by rhodopsin, and in turn it activated phosphodiesterase. In addition to the 3-part G proteins, there are other important G proteins. For now, we're going to discuss single protein GTPases that are key to regulating many intracellular processes. The best known is the RAS superfamily. Again, small GTPases which act as molecular switches. How does the switch work? Something in the environment of the cell changes. It could be a chemical or mechanical change that is sensed by a cell receptor. That triggers the G protein/GTPase switch, which in turn activates an effector molecule, which increases a particular cell process. The G protein that we are considering is a GTPase. When bound to GDP, it has a very low level of activity and doing little to stimulate the effector cascade. When a GEF, a guanine exchange factor, arrives, it causes the GDP to drop out. Because of the higher concentration of GTP in its environment, that is taken up in its place. Binding the GTP turns on the G protein activity to signal an effector molecule, which in turn increases the activity of a particular cellular function, like cell growth. But cellular switches should not stay on for long. The normal GTP hydrolysis reaction rate is very slow, but when it encounters a gap, a GTPase activating protein, that accelerates the hydrolysis of GTP back to GDP, and the G protein activity again slows down. So, holding on to a GDP means the switch is off. Arrival of the exchange factor with a change to GTP turns the switch on. Confusingly, the activating protein hydrolyzes the GTP, actually turning the G protein switch off. Got that? Now, let's see what we are switching. Regarding the small GTPases, there are 5 families. The initials come from the description of what they do. If you look carefully at their functions, you will see they regulate key intracellular processes. If this were a biology series, we would spend a long time here, but not today. I will mention one general example, and then one that relates to us. The RAS family, among many other things, controls cell proliferation, which is good when you want to grow. However, if the gap activating protein is mutated and doesn't work, that means the growth signal is always on. With the reproduction switch stuck in the on position, uncontrolled cell proliferation results in cancer. That was the story of the initial discovery of the RAS G proteins. To put it in a bigger picture, the widely quoted figure is that 1/4 of all human cancers have a mutant RAS family gene, making this a big area of cancer research. ARL, or ARF-like. Now we come to the member of the RAS family we are interested in. Unfortunately, the name ribosylation factor is a holdover from its initial discovery in the cholera toxin. Disregard that for now. ARL3 and ARL13 act together in intraflagellar transport. To relate that to the GTPase paradigm we were just discussing, the effectors of ARL3 are these carrier proteins which operate in ciliary transport. RP2 turns out to be the gap, the activating factor for ARL3. To complete the cycle, the GEF, the exchange factor, is ARL13. Other factors do play a role here, but this is the basic scheme. This shows you the layout of where the different actors are located on either side of the connecting cilium. Mutation in RP2 means it doesn't perform its function as a gap, and ARL3 is not deactivated. We know that this defect interferes with intraflagellar transport, ciliary integrity, and vesicle transport in the inner segment. So it must be a concentration-sensitive process. Since we are this far in, there are two other connections to mention. Mutations in ARL6 cause Bardet-Biedl syndrome, a prominent ciliopathy which we will discuss a little later. And mutation in ARL13B causes Joubert syndrome, having similar effects. Mutations of ARL3 have so far not been identified. ARL3 knockout mice show ciliopathy features and die very early. What a fascinating and important part of cellular biology. Before we finish, there are two syndromic varieties we need to mention. They are rare, but give us insight into important cellular processes. One is Bardet-Biedl syndrome. In addition to RP-type retinal damage, it turns out a gene mutation can also cause the following set of defects: obesity, kidney dysfunction, extra digits, and more. What could these various seemingly unrelated defects have in common? In the photoreceptor video, we discussed that most, that is, almost all, mammalian cells have cilia, a specialized projection from the cell which they use for different functions. One type is for motility, and the other is sensory. The BB-some is a multi-part protein complex that is partly responsible for organizing intraflagellar transport. At the base of the cilium, the BB-some assembles the IFT proteins, the motors, and the cargo for transport up and down the cilium. Here is an expanded concept view showing the major parts of the BB-some. If the BB-some is defective, then intraflagellar transport is impaired, and that affects a lot of things, making a family of diseases called ciliopathies. Affected organs include lungs, kidneys, retina, and more. Here are the identified genes. And if you wish to read more about ciliopathies and Bardet-Biedl syndrome. The other syndrome of special interest is Usher syndrome, which we mentioned earlier as the leading cause of autosomal recessive RP. We will consider this a little further now. There are about 40 syndromes in which vision and hearing loss are associated. Usher syndrome accounts for about half of them. In Usher syndrome, RP retinal changes are associated with loss of hearing and balance, divided into different categories based on severity, how early and to what degree each is affected. So how is it that a single gene defect can affect these two separate systems? What do they have in common? As you can see, the specific defective genes have been identified, causing each version of this syndrome. This is a schematic drawing of a hair cell, the ones inside the ear responsible for sound wave detection. I have simplified it to illustrate the concepts. The stereocilia protruding from the top have a core of actin filaments. When a sound wave arrives, the cilia are deflected. That pulls open a mechanical gate that allows ion flow and change of cellular charge. Here are most of the major Usher proteins, and in parenthesis, which version of Usher syndrome each is associated with. One group of proteins functions for cell-to-cell adhesion. Another group acts as a scaffold connecting the various proteins together. And the last one, myosin 7A, is a motor protein which carries the other proteins along actin filaments within the cell. The diagram shows one current theory of how the different proteins act together in the hair cells. Myosin is the motor that carries the harmanin and sans components from the cuticular plate along the actin filaments to the top of the cilia. Cadherin and protocadherin are cell-to-cell adhesion proteins that form the tip link, the connection between cilia, with the mechanotransduction channels on one end and the harmanin on the other end as the anchor scaffold. The harmanin end also seems to be responsible for maintaining tension in the tip links. Overall, this network of proteins is important in stereocilia formation, its ongoing function, and it plays a role in synaptic transmission. While researchers are getting a pretty good handle on the Usher proteins in the ear, they have a ways to go for the eye. They have found the locations of the Usher proteins, which is a start on figuring out what they do. The various proteins are found mostly at the connecting cilium and at the synapse. The first is thought to affect protein trafficking, perhaps controlling access through the connecting cilium, and the latter affecting signal transmission. In addition to the locations we just mentioned, if you watch the video on photoreceptor cell function, at the end we looked at myosin transport along actin filaments in RPE cells. The filaments are shown in red, with the myosin carrying melanosomes and phagosomes. The deeper we look, the more connections we seem to find. Here is one more connection that is just beginning to be explored. The sense of smell is mediated by a G-protein coupled receptor mechanism. In Usher patients, the same set of proteins we have been talking about have also been found in the olfactory apparatus. Implications for this are as of yet uncertain. One more point before we finish with Usher. Among the multiple causes of Usher syndrome, the most common cause of Usher type 1 is a defect in the myo7 gene. This has become a target for one version of gene therapy, which involves using an adenovirus to insert a normal copy of the myo7A gene. In this video on the biology of RP, we have only covered the headlines. The top 5 autosomal dominant mutations, though there are a lot more. The top 6 autosomal recessive mutations, though there are a whole lot more. And the top 2 X-linked mutations. We also took a look at 2 interesting diseases. One was Bardet-Biedl syndrome, based on a defective BB-some, causing a family of diseases called ciliopathies. The second was Usher syndrome, based on a functional group of proteins whose role in hearing is well worked out, but is still in process for the retina. With this video, we have tried to relate the many moving parts of the photoreceptor to the places things typically go awry, as seen in the family of retinal diseases that fall under the scope of RP. And that has taken us on an in-depth look at several parts of modern biology. Cancer may be the emperor of all maladies. It is more common than RP and has a diverse and interesting biology. Though RP is less common, it also has a diverse and interesting biology, and all its effects relate to a single cell type, the photoreceptor. So I would contend RP also belongs in the royal house of diseases. These references are selected for 2 reasons. One is they are good reviews, and 2 is because they represent the major authors in this field.